Good evening, everyone. So this crash course in molecular biology is organized by HiGenoMB, keeping in mind the important technologies to be for the students to be well aware of the important technologies in molecular biology. There are 8 sessions in this crash course, and the first session is on PCR and real-time PCR, which I will be taking today. I am Dr. Lakshmi. I am working as Deputy R&D Manager at HiGenoMB. I am leading validation and development and validation of PCR-based assays for infectious disease diagnostics. So the prerequisites of the webinar is, I believe the participants today have biology background and they know what is a living cell, what is DNA or RNA or nucleotide, and they know about Watson-Crick base pairing, that adenine pairs with thymine and guanine pairs with cytosine, and what is enzyme. So if you know these basic things, I will make sure that at the end of this session that you know the basic concepts of PCR. Okay, so the first question that might come to your mind is, why should we learn about PCR? So this PCR is a single technique which can give you a job opportunity in multiple applications. I am going to give you an example of how this PCR can be used in various applications. So this single technique can detect a single nucleotide change, which is widely known as mutation, or it can be used to detect a whole chromosome deletion or duplication, as you see in a genetic disorder. So apart from the diagnostics, it can also be used in other fields, such as when a gene has to be cloned into plasmid. So you all know about in 1990s or 2000s, there is a wave about cloning. So they cloned a sheep, the first cloned sheep called Dolly. So all of this is a very a small scale of cloning that happens in plasmid DNA inside a bacteria. So this can be done using PCR. And apart from that, so you people will be very well aware about COVID-19, and right now there is a surge of a new variant of COVID-19 infection that is spreading all over the world. And if you remember, in 2019 and 2020, for the diagnosis of COVID-19, real-time PCR was one of the technology which was widely used by all over the world. So apart from the disease diagnosis, PCR has multiple other applications in other fields, such as in food science. So there are few countries all over the world, that is in Europe, people do not entertain genetically modified food or genetically modified food items. So to identify those food items where the genetically modified food particles are used, to identify that, PCR can be used. Apart from plants, animal and food, it is also used in veterinary diagnostics. And in pharma industry, for environmental monitoring, PCR is one of the technique to identify if there is any contamination. Okay, now since we have established why this one single technique, if you know this one single technique, you will be industry ready. So this is the current trend, but what happens in the future? So there is a market research carried out by verified market research team, where they propose by 2033, this PCR application in medical application, the market will be valued up to 12.8 billion with a growth rate of 8.2%. And the market breakup would be it by the type of PC. If you are going to divide it by the type of PCR, standard PCR, real-time PCR, digital PCR, multiplex PCR, reverse transcription PCR. If you know any of these techniques, then your employability will really increase. And if you wonder in which field should I employ all this, you can go to infectious disease diagnostics as in a hospital setting, or genetic testing, or if you are interested in cancer research, blood screening and transplantation diagnostics. Okay, so now as we established why we should know about PCR, so what will this course cover? So we will cover the basic concepts of PCR, what are the PCR components and the functions, what are the 3 basic steps of PCR, the equipment and the types of PCR, mainly the real-time PCR and its uses. Okay, so we understood like many of our participants are from bachelor degree, and to cover the basics, so there will be lots of basic concepts that will be covered here, so that at the end of the session you will be clear about the concepts of PCR, the components used in PCR, and the reason why we are using all of this. Okay, so when first coming to what is PCR, it is a similar process that is happening in a living cell. So when you think of a living cell, the living cell reproduces itself by replicating the DNA or duplicating the DNA. So the same thing when it happens in a tube, and instead of one identical copy, now there are multiple, meaning millions and billions of copies are produced without a living cell, which is possible because of the invention of this revolutionary technique called PCR. So polymerase chain reaction is called in vitro amplification of DNA. Now we will see the difference between the in vivo DNA replication and PCR. So DNA replication happens inside a cell. It copies the entire genome. If you take a human cell, during DNA replication, there are a battery of enzymes such as helicase, primase, DNA polymerase, which is involved in a complex process, and it copies the entire genome into one more copy. That is, at the end of replication, there are 2 identical copies produced. And all of this occurs at the body temperature. When you take PCR, a segment of DNA is copied multiple times. And where it happens? It happens in a tube which is placed in a machine called thermal cycler. And here you use only a single enzyme which is called DNA polymerase. It involves 3 basic steps which I will be explaining it in the coming slides, and this is possible with a thermal cycler which varies the temperature between 95 degrees, 60 degrees and 72 degrees. Okay, so when we look at the history of PCR, where it started with the discovery of double helix structured DNA by Watson and Crick in the year 1953. So now they identified the DNA exists in the double helical structure. So the next thing is this DNA gets replicated or duplicated by an enzyme called DNA polymerase, which was discovered by Arthur Thornburg in the year 1956, and the paper got published in 1957. So this is one important component of PCR. The next important component of PCR is the primer. So this primer, this invention was made in the year 1971, where the single primer system and the 2 primer system was invented. Then in 1983, by Dr. Carey Mullis, the PCR was invented, meaning a basic version of the thermal cycler that we use nowadays was invented by Dr. Carey-Mullis. He showed that varying the temperatures at various temperatures like 95 degrees, at 60 degrees, at 72 degrees, if you vary the temperature and use appropriate enzymes and raw material, then a DNA can be copied into multiple copies, as in million or billion copies. But the challenge that he faced was because of the DNA polymerase. The DNA polymerase that was used in the olden days were not very thermostable, meaning in the different stages of PCR, the reaction goes up to 95 degrees or 60 degrees or 70 degrees. But the DNA polymerase that was used in the olden days were not thermostable, meaning after one cycle of PCR is done, they lose their activity. So because of this, after each cycle, a fresh DNA polymerase was added. Just imagine after each cycle, after 1 minute, you open a tube and keep on adding a component inside this. So this was painstakingly difficult, and it was very laborious. The whole scene changed when Taq DNA polymerase was incorporated into PCR in the year 1988 by a group of scientists at CETUS Corporation. So this Taq DNA polymerase has an advantage that it is a thermostable enzyme. It can withstand high temperatures. So introduction of this Taq DNA polymerase revolutionized the field, and now we have a plethora of PCR applications. Okay, so now let's go to the components of PCR. Okay, the first component is the template, meaning this is your gene of interest or the template of interest which you want to multiply into multiple copies in million or billion copies. Okay, so the generally used template is a genomic DNA which is isolated from various sources. It can be a human DNA which is isolated from blood, or it can be a plasmid DNA which is isolated from bacterial culture, or you can use complementary DNA which is derived from RNA. When you want to study about gene expression, you know the central dogma of molecular biology. The DNA gets transcribed to RNA, and this RNA is translated into protein. So now when you want to study the gene expression, then to study it at the RNA level, and RNA is not a very stable molecule, and it is difficult to manipulate RNA at the lab. To make it a bit sturdy molecule, a technique called reverse transcription is used, where the RNA is reverse transcribed by an enzyme called reverse transcriptase into DNA. So hence it is called complementary DNA. So we can conduct PCR with any of these three templates. That is a genomic DNA. It can be from any source, human or virus or bacteria, or it can be from, the template can be a plasmid DNA, or the template can be a complementary DNA which is generated as I explained now. Okay, so I just explained you how the introduction of Taq DNA polymerase revolutionized PCR. For this tremendous contribution by Taq DNA polymerase in the field of molecular biology, the Science magazine or the journal awarded Taq DNA polymerase as the molecule of the year in 1989. The success of the PCR as a technique which we see now was because of this discovery that Taq DNA polymerase can be used in PCR. So as I explained you, the very property of tagged DNA polymerase, that is the thermostability, was the reason for the success of PCR. And there are more variations which happened in the due course of time, and it improved the PCR overall as a technique. So this tag polymerase, although its activity is maximum at higher temperatures, say for example 60 degrees or 72 degrees, but at lower temperatures it had some intrinsic basal activity. Because of this, there was false or non-specific amplification. To avoid this, the scientists identified a technique called hot start, meaning the tagged DNA polymerase was encaptured in a antibody-like molecule, and this antibody molecule will get denatured when the reaction starts at 95 degrees. So you know the proteins cannot be stable at very high temperatures and they denature. So this technology was adopted and it was called hot start, and this discovery helped to curb the intrinsic activity of tagged polymerase at lower temperatures. Okay, the next property of tagged DNA polymerase is the processivity, meaning how many number of the nucleotides are incorporated in a growing strand of DNA in a single binding event. So this is called processivity, meaning when a tagged DNA polymerase binds to the template, it incorporates nucleotides into the growing strand and creates multiple DNA strands. So before falling off from the template, how many such new molecules it can generate. So this is called the processivity. And the next property of tagged DNA polymerase is its fidelity. So what do you mean by fidelity? Which means like it is error free. If 1000 base pairs are added by tagged DNA polymerase, there is a chance for one error per 1000 base pair. But in few assays or few scientific experiments, this errors is not admissible. To overcome that, high fidelity polymerases were included. So this is the next important component of PCR, which is called primer. What are primers? Primers are a short stretch of nucleotides. So if there is two nucleotides, we call this binucleotide or dinucleotide. If there are three nucleotides, we call them trinucleotides. When there are more than 5 or 6 nucleotides, when there is a stretch of nucleotide which is more than 5 or 6 base pair, then we call them as oligonucleotide. So these are the starting material for PCR. So these primers are required to locate the gene of interest in your template. So the points that are explained here are the basic points that has to be taken care while designing a primer. So your primer should be of length 18 to 22 base pair, wide not more and wide not less. So if the length is lesser, then there will be non-specificity. Say for example, the human genome is of 3.3 gigabases size, and I am going to use a primer of 5 nucleotides, say with a sequence saying AGCTG. So the occurrence of this AGCTG in 3 gigabase pair of the whole entire human genome is very high. Because of this, when I am using this primer in my PCR reaction, instead of amplifying the DNA or the gene that I want, I will be randomly amplifying anything and everything in the human genome, which is not my goal for the assay. So to increase the specificity, what we do, we are increasing the primer length. And what is the optimal length? So you can ask me why if the longer primer gives more specificity, why can't I use a very long nucleotide to have more specific product? The cost also comes into picture here. So 18 to 22 base pair is the optimal length which can serve the purpose. So the longer the oligonucleotide is not really required. Okay, so what is GC content? So in this 18 to 22 long nucleotide, the amount or the number of G and C nucleotide should be 40 to 60%. Say if I have 20 nucleotide long primer, at least 10 should have 10 of these nucleotides should be a G or C. Why are we more specific about GC content and not the AT? Because we know GC has the guanine nucleotide and this cytosine nucleotide are bound by has 3 hydrogen bonds for their pairing. Hence this GC content is important. Because of this GC content, this will increase your annealing temperature to 55 to 65 degrees. This is another reason why the specificity of your PCR reaction will be increased. And when you look at the sequence, the template sequence, avoid choosing those sequence which has a multiple runs of same nucleotide. Say for example, your template has a stretch of continuous runs of G, 5 times G, 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 G or C, C, C. So avoid those regions or avoid a repeat region. When I say repeat, so GCT or ATG, the same nucleotide is occurring in random, ATG, ATG, ATG in random, in continuous occurrence. So this is the difference between a repeat and a run. I repeat it again. A run is a stretch of nucleotide, is the same nucleotide which is repeated multiple times. Say for example, A multiple like occurring for 10 times in tandem. So this is called a run. A repeat is a stretch of nucleotide which is occurring in tandem. For example, ACT appearing in tandem. Okay, so these sequences should be avoided because when these sequences are present, it is difficult for the primers to find the specific regions and this will lead to non-specificity. Okay, so the next is avoiding self-dimers. So what are dimers? What are self-dimers? It means a forward primer molecule binding to another molecule of forward primer. So when these bind with each other, they are not available for binding to the template. So this will lead to lesser efficiency of PCR. And we should also, if you avoid these repeats, then we can avoid the secondary structures. If there are complementarity within the same nucleotide, then the nucleotide will tend to form secondary structure like hairpins or loops. So to avoid this, we should select a sequence which does not have a nucleotide which is complementary to the nucleotides present in the same stretch of primers. Okay, so we had covered template, DNA polymerase and primers. So what is the next component? It is DNTPs, which is deoxynucleotide triphosphates. These are the single units of bases, namely A, T, G and C. So as you all know, these are the building blocks of DNA, and this will be, when you say PCR, the process is all about incorporating these nucleotides into a growing strand of DNA as opposed to a template. And DNTP has another role that it provides energy for polymerization. Okay, so here this is an enzymatic process, right? PCR is an enzymatic process. So an enzyme to work properly or to work efficiently, it needs cofactors. So what are those cofactors? Magnesium and manganese. So this is required for the polymerase activity and also to provide specificity and fidelity for the process. Okay, apart from this, we use something called PCR buffer, and these are the general components used in PCR buffer. So the first component is tris HCl, which gives a pH of 7.8 to 8.5. This is necessary to prevent the degradation of DNA, and there will be a small amount of EDTA, because when I told in previous slide that magnesium is a cofactor that is required for polymerase activity. So if you all know, EDTA is a metal chelator, meaning it will quench the metal ions. So if magnesium is required for the reaction and you are adding EDTA, which is a metal chelator, how is it going to help the process? This EDTA is required in small quantity, because when there is unused magnesium ions, instead of enhancing the PCR reaction, these magnesium ions will inhibit the PCR reaction. So to avoid this, small amounts of EDTA is used to remove the excess magnesium ions. So apart from this, there are other reagents such as formamide, glycerol, and 7-DSR2' deoxyguanosine 5' triphosphate. This prevents the secondary structure formation. So in my previous slide, I told a primer should not form a self-dimer or should not form a secondary structure within itself, because the primer is required to bind to the template and not to itself. So when this is happening in the reaction, to avoid it, these components are added to prevent the secondary structure formation. So you know, formamide is a denaturant and it removes the hydrogen bonds that is formed between the base pairs. Okay, again these are all bivalent cations which are helpful in increasing the primer binding efficiency, and other components such as polyethylene glycol and gelatin are used to enhance the polymerase activity. Apart from this, some of the PCR buffers has DMSO. This is added as an additive to decrease melting temperatures. Okay, so we had seen the chemical components of PCR. To set the PCR in the lab, we need some plastic waste. So what are those? These are the PCR tubes. This is a 100 microliter or 0.1 ml PCR tube. This is a 200 microliter PCR tube. If you have limited number of reactions, then you can use PCR tubes. But if you are going to use a large number of reactions, then you use PCR plates, which has 96 such PCR tubes arranged in an array of 8 rows and 12 columns. So this is a PCR strip, and after you set the reaction, you use a sealer to cover the PCR plate. Okay, next we are coming to the real important hardware of PCR, which is called the thermal cycler or PCR machine. So what is a thermal cycler? It is an instrument that regulates temperatures in a cyclical program. I will explain about the cyclical program in the upcoming slides. Okay, what is this thermal cycler? What does it have? What is special about it? It's nothing but a heating block. So the heating block, it is made up of copper or silver, You would have studied in your 8th grade what is a good conductor of electricity, what is a bad conductor of electricity. So you all know copper is a good conductor of electricity, silver is a good conductor of electricity because these are all metals. So such a metal is used to make this heating block, and because we are going to rise and lower the temperature at different stages of PCR. Okay, there is an important quality about this thermal cyclus which is essential for the success of the PCR. So this is called ramp rate. So what is a ramp rate? How fast a thermal cycler can change between different temperature is called the ramp rate, and it is expressed in degrees Celsius per second. So I will tell you in the upcoming slide why is this feature important. So now you can see the basic schematic of a thermal cycler. It has a heating lid, it has an insulation block, it has a block called Peltier block, and there is a heat sink and a cooling fan. Okay, so this Peltier block is the device that is going to control the temperature of the block. How it performs? It performs using the electrical current that you are going to provide to the thermal cycler. So apart from this, you have an important component in the thermal cycler which is called heating lid. So I have been telling you that PCR is carried out at varying temperatures. The highest temperature that it goes is up to 95 degrees. So you all know when a liquid is heated to very high temperature, what happens? It evaporates. So if you are setting a reaction and your reaction gets evaporated, then what will we be able to see at the end of the reaction? Nothing, right? So to avoid this, to avoid the evaporation, there is a component in thermal cycler which is called heating lid. What it does, it increases the temperature of the PCR plate or your PCR tube to the lid of the PCR tube or the PCR plate will be maintained at high temperature all throughout your PCR reaction. Because of this, since the temperature at the external environment is higher, the evaporation is avoided. So this is the advantage or the use of heating lid in thermal cycler. Okay, so we saw the PCR components, the chemical components required for PCR. We saw what are the consumables. We saw about the PCR thermal cycler. So now we can learn about the basic steps of PCR. Okay, so this is the first step. So these are the chemical components of PCR which we saw now. Okay, so the chemical components and the PCR consumables we have seen. Now we will see what are the basic steps of PCR. Okay, the PCR reaction comprises of 30 to 40 cycles of 3 basic steps. What are those steps? So the first step is called denaturation. So here, if you take one copy of DNA At the step of denaturation, meaning you increase the temperature to 94 degrees. So what does this heat causes to the DNA? It breaks the hydrogen bonds that is formed between the two strands, the sense and antisense strand. So because of this, what happens? The two strands get separated. So now they exist as single strands. At this point, we are going to reduce the temperature from 95 degrees to annealing temperature, which is usually between 55 to 60 degrees. At this point, what happens? A primer anneals to the complementary bases of the template. Once this binding happens, the Taq DNA polymerase binds to the primer strand or primer template complex and uses the free 3' hydroxyl end of the primer to add nucleotides to the 3' end complementary to the bases present in the template. So this happens at 72 degrees for the time which is dependent upon the length of the amplicon that you require. So at the end of these 3 steps, which is called one cycle of PCR, you will have 2 copies. That is, one copy of DNA is denatured into two single strands. Then a primer is bound, which is essential for the Taq DNA polymerase to act upon, and at the end of this extension, 2 copies of DNA are formed. So these 3 steps are repeated in multiple cycles, like 30 to 40 cycles, and million copies of DNA is made. So this is the typical PCR program where denaturation for 15 seconds at 95 degrees, annealing of the primers happens at 55 degrees for 30 seconds, and extension happens at 72 degrees for 20 seconds, which is repeated for 30 to 40 cycles. So if you have started your assay with 1 copy of DNA, at the end of the 30th cycle, you would have made 1 billion copies of DNA, because after each cycle you are going to double the amount of DNA which was present at the initial stage. So you have to just do 2 to the power of the cycles that you are incorporating in PCR, which is for 1 copy of DNA, it becomes 1 billion copies. Okay, so now you have finished the PCR. You have your PCR tube done after your PCR run is over. So how do you see the result? How do I know if the PCR reaction was successful or not? So for this, you run a agarose gel electrophoresis, where you cast a gel which is made up of agarose. So this agarose has sieves or holes where the DNA molecule can migrate based on the size. This is enabled by using a buffer and an electric field. So here what you see is a DNA ladder or a marker which has bands based on the size. So this is a 100 base pair DNA ladder. The least band that is present at the end is a 100 base pair, and the one above it is a 200 base pair. So here we have a PCR amplicon of about size 150 base pair, which is fluorescing well under the UV light with the help of ethidium bromide. So this is called endpoint PCR, meaning you will get to know the result of the PCR after the whole reaction of 30 to 40 cycles is over, only when you load the PCR product on a gel, on an agarose gel. So this is called endpoint PCR. Okay, so if I am saying endpoint PCR, can I monitor the PCR run in real time? Yes, you can, but for that you need some modifications. So that we will learn about this in this part of the session, which is called types of PCR. So before we dive into the types of PCR, I want you to know some of the terminologies. One important terminology is called probe. So I have been talking about primers, and what is this probe? This probe is also like your primer, which is a single stranded DNA or RNA molecule. But what is the difference between a primer and a probe? It has dyes. It has a 5' fluorophore dye and a 3' quencher molecule. And these probes should also be specific to your template. Okay, these probes have fluorophores at their end, at their 5' or 3' end. So what are these fluorophores? These are the molecules that can absorb light energy through excitation, and it releases the energy at a longer wavelength by a phenomenon known as emission. So these fluorophores, the fluorescence emitted by these fluorophores will be quenched by a molecule called quencher. So these quenchers' job is to catch all the fluorescence that is being emitted by the fluorophore and do not allow any of this fluorescence to be emitted outside. So this is achieved by a technology called Foster resonance energy transfer technology. Here, when a fluorophore and a quencher are in close proximity, then the fluorescence emitted by the fluorophore will be captured or absorbed by a molecule called quencher. But when the quencher is away from the fluorophore, then this absorbance of energy by the quencher will not happen. Okay. So what are the types of PCR? There are multiple or a very wide variety of PCR available. Here I am going to give you the classification based on quantification. That is, either you can have a qualitative PCR, meaning you can detect if there is a presence or absence of a gene of interest, or you can quantify the amount of the gene of interest that is present in the experimental system that you are looking for. Based on the applications of PCR, there are plethora variants of PCR, but I am giving here 3 important variants of PCR. One is called colony PCR. If people know cloning, so how do you know when the plasmid that you have cloned has got the insert? So you have you would like to know if the plasmid DNA has been taken up by the bacteria, and if at all it has been taken up, does the plasmid DNA carries the insert that I had put into the plasmid. So to verify it quickly, people do colony PCR. So they pick the bacterial colonies, lyse it at high temperature, and use that as a template for the PCR for the insert that is inserted into the plasmid. So this is called colony PCR, which is widely used in cloning. And there is another PCR which is called asymmetric PCR, where instead of using 2 primers, you will use only single primer, where you will, at the end of the PCR, you will be multiplying only a single strand of DNA. So where is it applicable? Where is it useful? So it is useful in the generation of probes. So these are there are probes called hybridization probes, which has multiple uses, like southern blotting or northern blotting, or nowadays in next-gen sequencing you use hybridization probes to pull out gene of interest. So in all those things, this asymmetric PCR has its application. So the next variant of PCR is called the nested PCR. Here we use two sets of PCR, meaning we use an external set of PCR and do a few cycles of PCR, meaning 1 to 10 cycles or 1 to 20 cycles of PCR are carried out with one set of primers, where you have a longer strand synthesis happening. Then the rest of the PCR cycle, meaning from 21st cycle to the 40 cycle or the 30 cycle, you use a PCR you use primers which are within the external PCR primers. So in this case, you are increasing the specificity of your template. If you are using a clinical sample where you are looking for a pathogen which is not very abundantly present, so in that case, if you want to increase the abundance of the template, so in those cases you carry out nested PCR. So we saw a classification of PCR based on the quantification, right, which is called endpoint PCR or quantitative PCR. So we will see what are the differences between these two and where it can be used. So the main difference is based on their reduction. So if the reduction is post PCR, as I explained you, once the reaction is over, you take the PCR product and then load it on the agarose gel and then identify whether your PCR reaction is successful or not. So this is called endpoint PCR, for which you need a agarose gel. In real-time PCR, that is your quantitative PCR, you need fluorescent probes, and the detection is based on these probes. What is the purpose of endpoint PCR? It is only amplification. What is the purpose of real-time PCR? You can simultaneously amplify and also quantify, and there is no manipulation after PCR, meaning there is no labor post PCR. What about the speed or throughput? The speed is really slow in endpoint PCR because you have to cast a gel, you have to load the gel with the PCR products, but this is not required in real-time PCR. So the speed is really, and the throughput is really good in real-time PCR. And what about the resolution? What I mean by resolution is, if I have very lesser number of target in my sample, will I be able to, which is the best technique that I can use for low copy number templates? Endpoint PCR will not be much appropriate. Here real-time PCR gives gives a better resolution than endpoint PCR. Okay, what are the, is there any special components that to be added in real-time PCR? Yes, you need a fluorescent dye or probe. The end results in endpoint PCR is in the form of DNA bands on gel. In real-time PCR, the end results come as a melting curve or amplification graph, which we will be seeing in the upcoming slide. And when coming to the cost, endpoint PCR is relatively cheap, but the real-time PCR is costly because of the probes that you use. And multiplexing is possible in both, but has high accuracy in real-time PCR. Okay, so what are the applications of endpoint PCR? You can use endpoint PCR in cloning, in genotyping, and colony screening. And real-time PCR, if you have a gene of interest and you want to know the expression between two different conditions. In those cases, you can use real-time PCR. And if you want to quantify anything, if you want to quantify a viral load, then you can use real-time PCR. And real-time PCR is the go-to technique in clinical diagnostics. Okay, so I have been using this term real-time PCR. What is it? It's a PCR reaction where you are going to monitor the progress of amplification of DNA or cDNA in real time. And the monitoring is enabled by the use of fluorescent probes. So I just want to explain this with an example. So when a scientist has identified or discovered a drug which he proposes that it will kill the cancer cell line. And how does he studies it? He wants to show that when the drug is given, the normal cells are living happily, but the cancer cells are dying because this drug is going to attack the cell cycle machinery. The expression of one of the key proteins in cell cycle is going to be hampered by this drug. So this is the hypothesis built by the scientist, and he wants to study it. So he is going to use the real-time PCR technology for it. And how does he do that? He isolates RNA from both the normal cells and cancer cell line which has been treated with the drug, and then convert this RNA to cDNA, and then carries out real-time PCR with the primers that are specific to the gene that he is going to study, and then identify the difference of the gene expression. So here you can see a amplification curve. This is called an amplification curve of these two cell lines. So that these kind of differences is made possible because of the real-time PCR. Okay, so this is the next example. So you are well aware about the SARS-CoV-2 pathogen, which is used in the clinical diagnostics. So here, there are 3 different genes of SARS-CoV-2 which was targeted during clinical diagnostics, and all of these 3 genes can be amplification of all of these 3 genes can be happening at the same time in a single PCR reaction by the advent of this real-time PCR, and the results can be obtained. Okay, so all of this is made possible by a special component which is used in real-time PCR. What is the special component? It's a fluorescent dye or a fluorescent probe. So what is the difference between the fluorescent dye and the fluorescent probe? These fluorescent dyes are DNA binding dyes which will bind to DNA when they are double-stranded. So very well known example is cybergreen. And fluorescent probes, on the other hand, these are very specific to the target and design and synthesis this PCR probes like the same way that you do the primer designing. Here, when you order for this probes to your manufacturing company, you instruct the company to provide you what kind of probes that you would require. Like there are 3 different kinds of probes. One is called hydrolysis probe, the second type is called molecular beacons, and the third type is called dual hybridization probes. So here I am going to explain you about the DNA binding dyes, how it happens. Here the PCR reaction happens, as I explained you, in the 3 steps like denaturation, annealing and extension. So what will happen when such DNA binding dyes like cybergreen is added to the PCR tube in the PCR reaction? So when the cyber green is added to the PCR reaction, after the 3 stages of PCR is over, that is denaturation, annealing and extension, when the double stranded DNA is formed, the cyber green binds to the double stranded molecule, which will cause the fluorescence. So after each cycle is over, as and when there are multiple double stranded DNA is formed, there will be more incorporation of these DNA binding dyes. So as and when these DNA binding dyes bind to the DNA, there is lots of fluorescence which is emitted by these dyes. And after each cycle, the fluorescence will be recorded by the thermal cycler, for which a specific components are available in the thermal cycler. So a conventional PCR thermal cycler, which I explained you in the previous part of the session, cannot be used here. Here a special PCR thermal cycler which has special components, which I will be explaining in the later part of the session, will help in recording the fluorescence after the end of each cycle. So at the end of the first cycle, when there are 2 molecules of DNA are formed, if the fluorescence is recorded is 10, but at the end of the 30th cycle, the fluorescence will be exponentially increased, because the availability of double stranded DNA is high, and because of which the fluorescent binding dyes also emit lots of fluorescence after binding to this double stranded DNA. Okay, this is how a typical amplification curve appears after all the 40-45 cycles of PCR is over. So after DNA binding dyes, I was explaining about fluorophores or probes. So this is a probe called hydrolysis probe, where a stretch of oligonucleotide which has a 5' reporter moiety or reporter dye and a 3' puncture dye is attached to the short stretch of oligonucleotide. So let's see how this is employed in PCR. Okay, so this is stacked DNA polymerase. So this stacked DNA polymerase, what it does, it extends the primer molecule and produces a second strand, right? So this probe will be complementary to a template, and it lies between your forward primer and reverse primer. When the extension of the primer happens, the DNA polymerase will keep on extending the primer, and it reaches to the fluorophore or the probe. So here it gets stuck by the probe because it is being stopped by the probe. But what the polymerase does, it has an intrinsic activity called 5' to 3' exonuclease activity, which means it will chop off these fluorophores and releases the template for its synthesis activity to complete. Because of this action, the reporter gets released from the probe and it is free to fluoresce, and this fluorescence will get detected by the real-time PCR machine. Okay, so the one that I explained now uses the intrinsic activity of Taq polymerase, and it is called hydrolysis probes. We have 2 other kinds of probes, fluorophores, which is called molecular beacon and dual hybridization probes. Here, the molecular beacon has a unique structure, that is, it has a stem and loop structure. So what is the stem and loop structure? This is your oligonucleotide, which has at the 5' end as a stretch of nucleotide, which are complementary to the stretch of nucleotide at the 3' end. So these forms hydrogen bonds, and the loop does not have any complementarity, and hence it forms this structure called hairpin loop. But this loop has a stretch of nucleotide which are specific to the template that you want to amplify. So the next set of probes is called dual hybridization probe, where two set of probes are used, and this probe will have a donor, one probe has a donor fluorophore and another probe has an acceptor fluorophore. When they are in close proximity, the fluorescence emitted by the donor fluorophore will be accepted or quenched by the acceptor fluorophore. When the distance between the donor fluorophore and acceptor fluorophore increases, then the donor fluorophore is free to fluoresce, and the fluorescence gets recorded by the real-time PCR machine. Okay, let's see how this molecular beacon works. So when in the real-time PCR machine, so after each cycle, an electric current is passed and to excite the fluorophore molecule present at the 5' end. So this excitation will cause the fluorophore to fluoresce, but the quencher molecule, which is setting very proximal to the fluorophore, will not let the fluorescence out. It quenches all the fluorescence that is emitted by the fluorescent molecule, and hence no fluorescence will be formed. But when the loop structure here, which I told, will be complementary to the to your template, when this loop finds the complementary sequence in the template, it resolves this hairpin stem loop structure and it linearizes. Once it linearizes, the distance between the reporter and quencher increases, and when the electric current excites the reporter and when the fluorescence is emitted by the reporter, the quencher is not in very close proximity to the reporter, and hence the fluorescence is free to be emitted. And this technology is called fluorescence resonance energy transfer. So I kept on saying this excitation emission, and this is a table which is explaining you which are the commonly used fluorophores in real-time PCR and what is the excitation and emission maximum of these fluorophores. Okay, so we can compare the two different types of real-time PCR. So these DNA binding dyes is one kind of real-time PCR, and probes based real-time PCR, where I explained about hydrolysis probes, the molecular beacons, and the dual hybridization probes. So this forms the probes based real-time PCR. So let's compare the features between these two different types of real-time PCR. So as I told you, these cyber dyes can bind to any double stranded DNA. These are not specific. It does not care whether this double stranded DNA comes from your specific target. If the primer designing is not proper and the primer forms dimers with itself, then also double stranded molecules are formed. So this DNA binding dyes will bind to those primer dimers as well and release fluorescence. So in this way, this is not a very specific technique. On the other hand, if you take probes this kind of problem does not exist because you are designing a probe which is specific to the template, and hence the specificity is really high. And what about the sensitivity? Meaning, can I detect very low copy number of my target? Yes, that is possible when you use probe based real-time PCR, because you are using fluorophores here which are very sensitive and you can detect as low as 1 copy. So multiplexing, meaning in a single reaction, can I use multiple primers and detect multiple targets? Yes, that is possible in both, but with cyber based you should use special reagents called high resolution melting DNA polymerases. Then the cost is relatively low in cyber based real time PCR, since you are having fluorophore and other specialized reagents in the probe base. It's costlier compared to other types of PCR. So what are the applications of the cyber green? It is used widely for gene expression studies. It can be used for pathogen detection. In probe based real time PCR, when you are looking for a low copy targets, then probe based PCR is your go-to technique. And what are the disadvantages of cyber based real time PCR? As I explained you, there is possibility of false positives because the dye can bind to any double stranded DNA such as primer timers. The disadvantage of the probe based real time PCR would be the design of these probes are really cumbersome, and the optimization of probes is time consuming and it is expensive. If you are working in an industry and you require very high throughput technology, then probe based real time PCR would be helpful. Okay, so I had been telling you that there is a component in the thermal cycler which is used in real time PCR. So these are the components which are present in the real time PCR, which enables us to record the fluorescence emitted by the fluorophores or the DNA binding dyes. So the first component is the light source, which excites the fluorescent dye, and once it excites the fluorophore, then the fluorescence emitted by the dyes or the probe should be recorded or detected. So for this, there is a component called detector. What it does, it detects the specific wavelengths emitted by these fluorophores. There are different types of detectors which are used in different types of real-time PCR machines. At the end, we have a software which collects all these fluorescent data and converts that into a usable data and provides it to the end user in the form of amplification curves and threshold cycles. Okay, this is how a typical amplification curve appears after your real-time PCR. So here you have an initiation phase where at this stage the fluorescence has not increased to a substantial amount. So this lies in the as good as your negative control. But as the amplification proceeds, the fluorescence increases and the PCR reaction produces lots of fluorescence and it gets recorded, and the intensity of fluorescence increases with the increase of the cycle number. At some point of the PCR reaction, the raw material in the PCR reaction gets used up and hence no more PCR reaction happens and it reaches the plateau phase. So here you see a green color line, a dashed line, where the fluorescence is really well above the background. So the cycle number of the PCR where this increase, the substantial increase of fluorescence from the background, this cycle number is called the threshold cycle or quantification cycle. So I will explain you why is it important. Okay, so this when the fluorescence, when the PCR amplification happens, depending upon the starting material. Say for example, if you have 1 million copies of DNA as your starting material, the number of cycles for it to reach the fluorescence above the background fluorescence will be smaller, meaning for 1 million copies of DNA, when it gets amplified in a PCR reaction, after about 15 or 16 cycles, the fluorescence starts increasing above the background level, and it starts entering the exponential phase. But when you take a template which is of lower quantity, a 10-fold lower quantity, about 100,000 copies, for this 100,000 copies to reach this exponential phase, it takes some more number of cycles, probably 3 or 4 more cycles. So as you can see, based on my initial copy number, the threshold cycle increases. So which gives you the interpretation that the amount of DNA, initial amount of DNA is inversely proportional to the threshold cycle. Okay, where is this useful? This is useful when do you want to do a absolute quantification of the gene of interest. So the CT cycles that we obtained from this, from the dilution series of your template DNA, we can plot it against the log amount of DNA and the CT cycle to obtain a standard curve. So this we will obtain from the standards. What are the standards? The standards are already known copies of DNA which you will use in the PCR reaction, and then use your unknown sample. So for your unknown sample also, you will arrive at some CT value. So when you plot the CT value to the standard curve, then you obtain a concentration of your unknown. So this will help you to determine the copy number or the viral load in of the sample of your interest. So if a patient is undergoing a treatment and the doctor wants to know if this treatment is working, he wants to know if the viral infection that he is treating is decreasing as he gives the treatment. So for this, he will do a absolute quantification assay, or the lab will do an absolute quantification assay to identify how the viral load is decreasing upon the success of the treatment. Okay, there is another technique or another method called relative quantification, where you can quantify the amount of your target in relation to the control gene. So this control gene, the property of your control gene should be that it cannot vary between various conditions. Meaning, if you have a cancer tissue and a normal tissue, you want to study the expression of a particular gene interest, a particular gene expression difference between cancer tissue and a normal tissue. To study that, I am going to select a I am going to select a control gene. With reference to this control gene, I will study the target gene. So how will I select the control gene? The control gene should be a housekeeping gene which will not vary its expression in different conditions, meaning in cancer tissue and also in the normal tissue, the expression of this gene is maintained at the same level. As an example, this can be your cell cycle machinery, or it can be your cytoskeleton proteins like beta actin, which will not change in cancer tissue or the normal tissue. So in relation to this housekeeping gene, you study the gene expression of your interest and compare the differences in the two different tissues that you are interested in. So this is widely used in the expression level of target gene in drug response studies. Okay, so with this we will conclude this seminar. I hope this seminar is informative